An announcement. Some students have been asking about the tutorials. They are going to be launched this week. There's been a number of technical issues between Achieve and Brightspace and coordination and getting content uploaded properly. I just talked to Dr. Pemberton, and that has mostly been resolved, and we're just going to do a test launch to make sure everything's working. But the goal is that in the next day or so that the tutorials will be available, the content will be available. The quiz will probably be pushed into February just because there's been so many delays in getting this content up and running. So it will happen. I apologize for the delay, but again, learning curve for everyone between Achieve and Brightspace. So I think we've finally got it sorted out. So today we're going to talk about receptor-mediated endocytosis and a unique process called autophagy. So we'll focus on how do you get stuff from the outside of the cell to the inside of the cell in a sort of specific, guided, coordinated manner. There's many types of forms of receptor-mediated endocytosis. We're only going to focus on one, and that's the low-density lipoprotein, which involves really important cholesterol metabolism. And then we'll talk about this unique way of getting rid of bulky, problematic material found inside of the cells in the form of maybe large aggregated proteins or damaged organelles like mitochondria. So we have the last week we talked, or the last couple of weeks we talked a lot about how we get proteins from the endoplasmic reticulum to the Golgi and how they can be sorted either to go to the lysosome or they can be secreted. Today we're going to talk about the opposite way, is getting stuff from the outside of the cell inside, and how we can direct some of this extracellular proteins to the lysosome. But before I get into that is not the only way we can get stuff from the outside to the inside. So phagocytosis generally refers to generally a non-specific engulfment of material outside of the cell. The best example would be phagocytic cells like macrophages that are able to engulf things like whole bacteria and cause their destruction. So that's phagocytosis, engulfing a large thing like a bacterium and ingesting it and then destroying it. Penocytosis is basically little involutions of the plasma membrane, which then branch off. So it's almost like sipping water, like sipping a little bit of the contents outside of the cell. And they form these little tiny vesicles that go inside of the cell. So that's penocytosis. It's not specific. Cells will just occasionally cause these little involutions and buddings of the plasma membranes, little tiny amounts, and some of the contents from the outside of the cell get taken inside, but there's no real guided way of doing that. Today we're going to talk about receptor-mediated endocytosis. So this is the selective internalization of specific extracellular molecules, usually proteins. It could be other things though. And we're going to talk about the low-density lipoprotein today, but there's other molecules. In the past we used to do a whole lecture, incorporate transferrin. Kind of works on the same principle. There's a couple of little subtle nuances. I took it out just because I thought it's just more stuff to remember that isn't really that critical. So we'll only focus on LDL and how it gets internalized. But you should be aware there are other proteins that can be specifically internalized. So transferrin is one, hormones like insulin, and certain glycoproteins. So I'm going to go through the steps. I'm going to give you just a sort of a broader overview. So when we're looking at proteins found on the outside of the cell, they have to get inside. And we want this to happen in a guided manner. So we have a thing like a protein, for example. And this is soluble, free-floating on the outside. And so it's going to bind to a receptor. And then that's going to cause a recruitment of certain types of proteins, in this case, adapter protein. So we talked about that, and then also associated with that will be clathrin. So we talked about this when we looked at proteins that are destined for the lysosome. Very similar in that you have adapter protein, adapter coproteins, and clathrin. There's just a slightly different flavor. So what we talked about last week was AP1, those adapter protein 1. Those form at the trans-Golgi network, at the top of the Golgi, and that also causes the recruitment of clathrin. Now, so something very similar happens at the plasma membrane when a ligand, i.e. a free-floating protein, binds to its receptor, is it's going to cause a recruitment of these coproteins. In this case, it's AP2. PN stands for plasma membrane. So those adapter proteins, AP2 proteins at the plasma membrane, will associate in part triggered by, in this case, a ligand, protein binding to its receptor. We'll talk about a little bit more of the mechanism in the next couple of slides. But this recruitment of these coproteins and again co-recruitment of the clathrin proteins causes that invagination. Puckering, I don't know what you want to call it. Invagination is the proper term. So the membrane kind of folds inward. And then it is also going to require the closure of the membrane here to form that vesicle. This requires a protein called dynamin. So it basically constricts and constricts and squeezes and squeezes and squeezes and then pinches the neck so much so that it forms a complete membrane. And again, that requires energy in the form of GTP hydrolysis. What's shown down here is a transmission electron microscope image. I'm going to learn about that once you get access to the Achieve tutorials. Dr. Pemberton is going to talk about transmission electron microscopy. So in this case, it shows this invagination of the membrane, and that those little black dots are actually antibodies that are selectively bound to dynamin. And so you can see the dynamin is associated with the neck, and that's showing that it's actually pinching off and forming that, what will ultimately be called a clathrin-coated vesicle. So clathrin is on the outside, but then remember there's also adapter proteins. It's a complex of these things. So the take-home message here is that the ligand binding to the receptor is going to cause the recruitment of adapter proteins, AP2. That's also going to cause the co-recruitment of clathrin, and those two proteins together are going to cause that deformation of the plasma membrane. It involutes, vaginates, if you want to call it that. And then what's the other thing is that you need the dynamine and GTP hydrolysis to squeeze that, that neck, so to speak, so that it will form this complete clathrin-coated vesicle. Okay, so that's kind of just generally talking about receptor-mediated endocytosis, the first stage. Let's focus on a specific molecule, so low-density lipoprotein. So lipids don't really like kind of floating around in our circulatory system. They tend to get encapsulated in these things called low-density lipoproteins. And so right in the middle there would be cholesterol. So we all know about cholesterol. Generally, when you get to my age, you worry about it a lot because it can clog your arteries. And so this cholesterol, it doesn't generally free flow. So it gets encapsulated in these lipoprotein molecules. So the lipids are basically encapsulated by this these cholesterol fats, so to speak, encapsulated in phospholipids. So the inner part here is hydrophobic, because that will face the fats. Fats are generally hydrophobic. And then it has a polar surface. So there's phosphate groups linked to the lipids. So that's what hence the name phospholipid. So that is polar. So that has a charge and it can interact with water because it's hydrophilic. The inner portion, so these hydrocarbon chains here on this sort of phospholipid, and of course the big goopy butterball, as I like to call it, lipid, or fat in the middle, is hydrophobic. So this is what we refer as an antipathic situation. So the phospholipid monolayer that's encapsulating this as polar surface that faces into the outside, it's in your bloodstream, so that's hydrophilic. And then it also has hydrophobic center. So that's what's going to interact with the cholesterol esters. So LDL is basically about, let's just say about almost 90% of your cholesterol is going to be encapsulated in these things I call them big butterballs. So the inner part is the butter, and the outer part is a sort of a charged lipid, phospholipid monolayer. And so this is important for encapsulating your cholesterol and into this sort of large ball structure, and then it's going to be targeted towards LDL receptors. How is it going to do that? Well, there's a protein that encapsulates around the ball. It's called apolipoprotein B. So I call it the belt that wraps around the butterball. And so that apolipoprotein B is going to be what's going to facilitate the interaction with the LDL receptor. And so those LDL receptors are found in the plasma membrane, and we quite frequently, when we stain, we can see that they are associated with what we call clathrin-coated pits. I'll explain that in the next slide. So again, this notion, remember last week I talked about how proteins can bind to other proteins at certain pHs, and other pHs they don't bind. This is another example of that. So in this case, when we look at the LDL receptor, it has a couple of features. So here's the plasma membrane. The LDL receptor has a transmembrane domain. It also has a small portion that faces into the cytoplasm. And that is important for recruiting adaptor proteins, which I'll talk about in a minute. But the other important thing is it has this arm. It's a ligand binding arm. So this is the outside of the cell. Here's the plasma membrane, and there's the cytoplasm. And so at roughly neutral pH, between 7 and 7.4, the extracellular environment, that the ligand binding arm will bind very efficiently to the apolipoprotein B. So remember the belt around the butterball? So there's the LDL particle, there's the belt, that's the apolipoprotein B, it's wrapped around it, and that will have a high affinity for the ligand binding arm of the LDL receptor. And this will occur at the roughly neutral pH of the cell surface. So at pH 7-ish, it has a high affinity. So the ligand binding arm is going to wrap around the belt and hold on to it. And so that is going to cause a conformational change in this receptor, particularly at the part that faces into the cytoplasm. So it's got a sorting signal. signal. We've talked about a lot of sorting signals. This is one more. It's called the NPXY sorting signal. And that will cause recruitment of the AP2 co-proteins. And that will eventually lead to the formation of a clathrin-coated pit, which I'll get to in just a minute. So the concept here is really to talk about the idea of pH and how pH regulates binding. So at the neutral pH found at the cell surface, the LDL receptor ligand binding arm will wrap around the apolipoprotein B, and then that's going to trigger the sorting signal to cause the recruitment of the AP2 co-proteins and start to cause that involution or invagination of the membrane to start to form a clathrin-coated pit. Once you get the formation of the clathrin-coated pit, and ultimately it's going to take off the coat and it's going to fuse with an endosome or form an endosome and fuse with a lysosome, which I'll get to in a minute. So don't worry about those steps too much right now. Don't take notes on that just yet. It's important to note when it gets to the endosome is the pH is going to be different. It's going to be lower. So at a low pH, what happens is there's this thing called a beta propeller domain. Okay, so it's kind of hard because I have to hold a bunch of things. So here's my ligand binding arm. Let's say right my armpit is my beta propeller domain. And at the lower pH in the endosome, what happens is there's these histidine residues that get protonated. So protons will bind to the beta propeller domain. So right here, this is my beta propeller domain. And at that low pH, what happens is that causes a positive charge to be put onto the beta propeller domain, and then the ligand binding arm now has a high affinity. So it wants to be attracted. So let's imagine my arm is out here at neutral pH at the cell surface, grabbing onto the apolipoprotein B, the big butterball, and we internalize it, move along into the endosome. Endosome gets pH drops, start to get that protonation here in my armpit, and my arm swings down because it has a high affinity for the beta propeller domain, and poof, I drop the ball. So that's basically how it works, is that understanding that the pH dynamics determine how the ligand binding arm will bind at the neutral pH of the cell surface, but it won't bind to the LDL particle in the endosome because it's acidified in the endosome. I'll talk about how that happens in the next slide. This causes protons to be attracted to the beta propeller domain, so now it has a positive charge, and now it has a very high affinity for the ligand binding arm. Causes it to swing inwards and release the LDL particle into the lumen of the endosome. So I'll talk again. Now let's kind of introduce the concepts. Now let's put the pieces together in the order in which they occur. So we have an LDL particle floating in our bloodstream. It's got that apolipoprotein B right around the big butterball. That's going to bind to the ligand binding arm of the LDL receptor. Why does that happen? Because it's the neutral pH. Okay, so that's the first step. The second step is the NPX sorting signal is going to be now, it's basically you bind onto the butterball and it causes a little jiggering around, and my leg is facing the cytosol, my toe points out, there's an NPX sorting signal, AP2 co-protein coming over and bind. When it does that, it's going to cause a deformation of the plasma membrane. It's going to start to involute. It's also going to cause the recruitment of clathrin. So together between the AP2 co-proteins and clathrin, it causes the conformational change of the plasma membrane. Loops inwards. Okay, so the key here is, remember the pH, ligand binding arm, binding to the apolipoprotein B. This is neutral pH. The next step causes a little signal, NPX sorting signal. The blue thing there is your AP2 co-protein, and that will also cause a recruitment of clathrin. This will now start to form what's called a clathrin-coated pit. Now, I mentioned that the endosome can be acidified. Why is it acidified? Because there is the internalization of these things called B-class proton pumps. And so what they do is they can transport hydrogen ions from the outside of the cytosol into the lumen of the endosome. And these are also found in lysosomes, by the way. There's also, so this takes energy. ATP hydrolysis helps pump protons from the cytosol into the lumen. There's also chloride channels, and once the proton ions start to accumulate, there's a passive movement of chloride ions. So this, basically it's called creating hydrochloric acid. It creates an acidic environment in the lumen. So these proton pumps and chloride channels are found on the membranes of the endosome and lysosomes, and they're primarily responsible for causing the inside of these structures to be acidic. Okay, so same thing. We talked about this part up here, about the binding of the LDL particle via the apolipoprotein B to the LDL receptor or via the ligand binding arm. We talked about those co-proteins. This is our clathrin-coated pit. We find that at the plasma membrane. It's not shown here, but remember dynamine will be involved in GTP hydrolysis. So that's going to cause the pinching off of that membrane. Now we have a clathrin-coated vessel. You've got to get co-proteins off. Remember, if anything has to fuse, you have to remove these co-proteins. So at this point, once you remove the co-proteins, it's now referred to as an early endosome. There will be the insertion of those V-class proton pumps and chloride ion channels. So this will now start to promote the acidification of this early endosome. It matures. Now there's this, I get too bent on endosome. The endosome, late endosome, exosome dynamics are very, very, very complex. And there's a lot of fusion and breaking apart of these types of structures. I don't even want to touch that because it's super, super complex, so much so that I don't even understand it. I don't expect you to either. What's important to note is that the early endosome can mature or fuse with other endosomal structures to form a late endosome. That's as much as I know, that's as much as I care you to know. And at this point, you're going to have, at this stage, you're going to have, once it matures to the late endosome, the buildup of those proton pumps. So now you get a much more acidic environment. So at this stage, what happens to the LDL receptor? The beta propeller domain gets protonated because you've got all those hydrogen ions. It's got a positive charge now, and it's going to cause the ligand binding arm to swing down and release the LDL particle. And so this is at more acidic pH, pH 5. And then that endosome can go on and fuse with the lysosome, and then the cholesterol, because of the acidic hydrolases that are found in there, will degrade the big butterball and down to its individual fatty acids, which can be released and used for beneficial things like lipid synthesis. And what frequently happens is the late endosome can also pinch back off and form I don't even want to use the structure. Basically, the late endosome can go back up and it can fuse with the plasma membrane and recycle the LDL particle. And so at this point, when it gets back up to the plasma membrane, it's now neutral pH, and then boom, out pops the arm. It's no longer attracted to the beta propeller domain because you're now at neutral pH, and it's ready to find another LDL particle. So this can go on and on many times. And so it's believed that approximately 50% of the plasma membrane is internalized, like if we looked at skin fibroblasts, by receptor-mediated endocytosis every hour. So your membranes, plasma membranes are internalized. They're going through this process. Some of it will be directed towards lysosomes. Some of it will be recycled back up to the plasma membrane, fuse back with the plasma membrane, and restore those receptors back up there so then they can go on and continue their function. This happens fairly quickly. It's believed to take about 10 minutes. That's pretty fast for all these things to happen and to recycle back up to the into the membrane. Okay, so why is this important? People have probably heard about, no, I'm going to watch my cholesterol. Well, normally not do so much because you're young, but when you get old like me, you do look at it. My cholesterol is too high, and my doctor brags on me about it. So, but there are situations where people have genetic deficiencies. For example, their LDL receptors. So either they make no receptors, or the receptors can't bind to LDL properly, or the receptor can't internalize. So think about it. When I mention these things, I could put test question. You could say, hey, if you mutated the beta propeller domain, what would happen? If you mutated the ligand binding iron, what would happen? If you mutated the MPX sorting signal, what would happen? That's basically what this is saying, but I'm giving you more specific examples to understand the mechanism. Those things happen, and these things in this inherited form of what's called hypercholesterolemia. Familial means inherited, hypercholesterolemia means you have lots of cholesterol. So it's circulating throughout the bloodstream, and you're not able to internalize it. And so that cholesterol can build up in your arteries and form things called plaques. It can make your arteries much more stiffer, so it can increase your blood pressure. It can also start to accumulate and form fairly big chunky deposits, and that can actually block blood flow, which could lead to things like a stroke or a heart attack. And quite frequently, people that have familial hypercholesterolemia, they have a couple things that are not so serious immediately, which is xanthoma. It's basically the accumulation of lipids. You can actually see it, they form bulges. For some reason, it can accumulate at the elbows. But more importantly is that you're getting these plaques, stiffening of the arteries, and atherosclerotic plaques, cholesterol-containing plaques that are building up in the arteries, and that in turn can trigger a heart attack, because if it's an artery, like a coronary artery, which feeds blood flow to your heart directly, and your heart doesn't have sufficient blood, that can trigger a heart attack. Or if it's a vessel that feeds, a major vessel that feeds blood to your brain is deprived of oxygen, and that, if it's a part of the brain, for example, that controls movement, or any other thing, can lead to what we call a stroke. So this happens to people in their late 20s, where those type of things might happen to people normally with age, but maybe in their 60s, 70s, 80s, or 90s. Anyway. Oh, by the way, this is sort of leftover from a previous instructor, kind of like putting up Nobel Prize laureates. I'm never going to test you on who won the Nobel Prize. I think it's just important to show that understanding basic biology led to Nobel Prize. How does this begin? Okay, so now we're going to move on to a sort of a strange process called, I call it autophagy. Some people call it autophagy. It's not autophagy. I like to call it autophagy. But some people call it autophagy. I don't know why. I got interested in this over 20 years ago when I was working as a postdoc in the Salk Institute in San Diego. I didn't know much about it, and I got involved in a project working on it, and I learned to develop an appreciation for it. Yes, this picture is disturbing. Basically, it's a pig that's cutting himself off. The only thing that's missing is if he's shoving his entrails back in his mouth. So literally, it's self-eating. Depends on the lysosome. And because remember we talked about lysosomes have those acidic hydrolases, really good at breaking down lipids and proteins. And so this is a way to selectively get rid of potentially problematic junk in your cells. And those could be things like long-lived proteins that start to aggregate and form problems, or damaged organelles like mitochondria or peroxisomes. And that, I'll talk about in a minute about why those could be problematic. So generally, big, bulky things can be degraded through this lysosomal system. And we now know that if there's deficiencies in this, it's implicated in aging, and also perhaps in diseases, either age-associated diseases, or now they're starting to identify diseases in which there actually are deficiencies in autophagy. So it basically works like this. You have certain proteins. So we'll give in any particular cell 10,000 plus different types of proteins. And they go on and they do their function. Some proteins have a very short half-life. When we talk about cell cycle and cyclins, they have a relatively short half-life. They don't persist for very long. And there's a way to get rid of them through what's called the ubiquitin proteasome system, which I'll talk about in a minute. Other proteins can persist for days, weeks, years. And the longer a protein persists, the more likely it can misfold or have modifications to it that no longer make that protein healthy or functional for the cell. So if they form these sort of large misfolded aggregate proteins, the easiest way to get rid of them is to direct them towards the lysosome and they can be chewed up. The problem is, how do you get big bulky proteins
pass a membrane, so this is a lysosome, the answer is it's not easy. But discoveries actually made in yeast identified a whole bunch of proteins called autophagy genes, or ATG. And when they looked at these proteins they discovered in yeast, they found they're highly conserved. We find pretty well the exact same proteins found all the way up to mammals, including humans. And so what these proteins do is they help form initially, so here's our problematic material, let's say some protein aggregates, those little squiggly lines. And so they will be sort of targeted with what's called an isolation membrane, also known as a phagophore. Now, in the next slide, I'll talk about that a little bit more. So this isolation membrane, A, has to be guided towards this problematic piece of cellular debris, and then it extends around it. So it's this sort of double membrane structure. It's like a little C structure that starts to form around that problematic material. So there's a number of proteins involved in starting this process. ATG 12, 5, 16, there's like a couple dozen of them, right? I'm just mentioning it. I'll talk a little bit more about ATG 5, but this complex of ATGs are important for initially starting the formation of that isolation membrane. So they're kind of involved in the early steps of autophagy. In order for that membrane to extend, it needs help in the form of a protein called ATG8. ATG8, it was identified through a whole different process independent of autophagy. So in the literature, they switch back and forth between ATG8 and LC3. They are both the same thing. And ATG8 can bind a little bit of lipid. And it can recruit that lipid up to this isolation membrane and help extend it. And so ATG8 helps extend this membrane, doubles membrane all around the problematic material. Then, that double membrane structure, which is called an autophagosome, will now fuse with a lysosome, at which point it's called an autophagolysosome. The next slide, I'll reiterate this. Don't worry if you missed something. And at this point, the contents of the lumen of the autophagosome come into the lysosome, lumen of the lysosome, or the autophagolysosome, and it can break down those proteins into individual amino acids. And those individual amino acids can be recycled and used to make new proteins or other things. On the left, we can have a thing like a damaged mitochondria. And that mitochondria, damaged mitochondria, or mitochondria that are not functioning very well, can produce harmful molecules called reactive oxygen species. And if they build up to any great extent, they can cause a lot of damage to cells, to membranes, to proteins, to nucleic acids. So ROS is actually a driver of many disease processes and also the aging process itself. So mitochondria are one of the major sources of reactive oxygen species. So we talked about electron transport, the electron transport chain. Sometimes electrons kind of fall off, they don't get to the end of the road, and they cause a partial reduction of oxygen, and it forms these things called superoxide or reactive oxygen. Just call it ROS, R-O-S, reactive oxygen species. You don't have to get into the details. There's many different flavors. ROS can cause a lot of problems inside of cells. Now, I mentioned that you can have short-lived proteins that can be degraded, not through autophagy, but through another process called the ubiquitin proteasome system. And it basically works like this. So we have a protein that we want to get rid of. It hasn't necessarily misfolded, but it served its function. When we talk about cell cycle regulation, cyclins in the next lecture, this has a lot of relevance to that. And so this target protein can be recognized by a protein called an E3 ubiquitin ligase. So it has a binding site for the target protein, and it also has a binding site for what's called an E2 ubiquitin conjugating enzyme. So what it does is, one arm binds onto the target protein, the other arm binds onto the ligase. That's the thing that actually sticks on ubiquitin. What is ubiquitin? It's a very short protein. It's 76 amino acids long. And basically, it can be added onto certain lysine residues of the target protein. And it can be added on as a chain. It's called polyubiquitination. When that protein is polyubiquitinated, it will be released from the E3 ubiquitin ligase and then directed over towards what's called the proteasome, the 26X proteasome. It's a large, complex structure. It's like a garbage can almost. It has a lid that opens up. And in you shoot in the target protein, and as it's going in, you actually remove the ubiquitin polypeptides. That takes a little bit of energy in the form of ATP hydrolysis. So you're not degrading the ubiquitin, you're simply taking it off using ATP hydrolysis to provide energy for that. And the target protein is going to go into the barrel, it's filled with certain enzymes that chew it all up into pieces, and then you have a degraded protein, and the ubiquitin molecules can be recycled. And then they can be used to be conjugated to the next target protein. So this was actually one of the sort of earliest discoveries about protein turnover was via the ubiquitin proteasome system. Now, as we move forward and we start to discover there's other ways to get rid of proteins, and autophagy plays a role, and there's also ways to get rid of damaged mitochondria, and then mitophagy plays a role. There's a little bit of crosstalk, interplay between ubiquitination and autophagy. So this leads to how do we get rid of mitochondria? So the degradation of mitochondria using autophagosome is actually a process they call mitophagy. So it's autophagic clearance of mitochondria. This is, I tried to find that simplest diagram I could. It is way more complex than this, but I'm going to try to just talk about two major players, pink and parkin. Now, these two proteins were actually initially identified in screening for people that have an inherited form of Parkinson's disease. So Parkinson's disease is a brain disorder, a neurodegenerative disorder. It's loss of neurons. In this case, it's a part of the brain called the substantia nigra, and it produces these neurons that produce dopamine. And then that part of the brain is really important for controlling or coordinating movement. And so there's a rare individuals, actually there's a lot of different genes that have been identified that cause inherited forms of Parkinson's disease. These are just two of them. So the gene that encodes pink and parkin, when it's mutated and they no longer form function, they have no longer a functional protein, that leads to the degeneration of these neurons in the brain. And Parkinson's patients, usually they get what's referred to as early onset. Most people that have sporadic Parkinson's with no clear familial history usually get it 60s, 70s, 80s. These people could get it in their 40s or 50s. And so, well, what does pink and parkin do? Well, pink one is a kinase. It puts phosphate groups on certain proteins. That's what kinases do. When we get into signal transduction, we'll talk a lot about kinases. And parkin is an E3 ubiquitin ligase. So just like I talked about previously, that helps facilitate putting ubiquitin molecules on target proteins. So how does this work? So there's some kind of signal in that mitochondria is not functioning well. When we talk about apoptosis, one of the things about a sick mitochondria that can potentially trigger apoptosis is called mitochondrial depolarization. Remember that electrochemical gradient that builds up in the intramembrane space? It can start to dissipate. That can either trigger apoptosis, that's what that delta psi thing is. So that electrochemical gradient is starting to wane. The electron transport chain is not working so well. It's probably producing ROS. And if it continues to produce high levels of ROS, it's going to cause damage. And it seems to be much more problematic in the brain. Neurons do not take well to mitochondria that are not functioning producing ROS. They're very selectively vulnerable, and they can die off. And that's what's happening in this case in Parkinson's disease, is that the neurons within the substantia nigra are starting to have very dysfunctional mitochondria, and that are producing ROS and triggering apoptosis. And when that happens in normal individuals that have wild-type versions of pink and parkin, what happens is the pink 1 kinase gets stabilized and recruited. So normally it's degraded through a complex mechanism, which I will not talk about. but it gets stabilized and it binds to the outer membrane, usually through Tom proteins. And when it binds there, it now will actually phosphorylate parkin, the E3 ubiquitin ligase. And the phosphorylation of parkin will lead to its activation. So now its E3 ubiquitin ligase activity will be increased. And so it will now start to ubiquitinate substrate for target proteins found on the outer mitochondrial membrane. For example, it can now start to ubiquitinate mitofusion 1 or 2, which we talked about last lecture, or the VDAC, that's that major porin. And so that those polyubiquitinated molecules will attract LC3 or ATG8. So remember I talked about ATG8? I said sometimes they call it LC3. So the polyubiquitination acts as a trigger to cause the recruitment of LC3. There's a bunch of adapter proteins involved in it. I'm not going to test you on that. It's not important. Just know that in order to start autophagy, you have to have the recruitment of PINK1 to the outer mitochondrial membrane via TOMS. That in turn will cause the phosphorylation and recruitment of parkin. So it helps recruit parkin, but it also activates it. So it will now start to polyubiquitinate proteins found on the outer mitochondrial membrane. I'll give you 3, there's many more, but mitofusins and VDACs. That polyubiquitination will act as a trigger to cause the recruitment of LC3, also known as ATG8. LC3 has a lipid thing that can actually bind to the phagophore, or the isolation membrane. Remember I talked about that in the last lecture. So what that does is you get more LC3 binding to the polyubiquitination, and then also bringing in the isolation membrane and extending it. So now LC3 extends the isolation membrane, or the phagophore, all the way around the mitochondria to form the autophagosome. That in turn confused with a lysosome to form an autophagolysosome, and that will promote the degradation of mitochondria. So there's an example of initially screening to find disease-associated genes like PINK and parkin. And just like I talked to you about in my first lecture, figuring out how these proteins work. When they first discovered these genes, and then tried to say, okay, well, it's a kinase, I don't know what it does. And what's parkin? It's an E3 ubiquitin ligase. It did not immediately pop out. That was the function. They had to manipulate expression of these genes in a cell to see, hey, they're associating with mitochondria, not just any mitochondria that are damaged. And so one of the emerging themes in Parkinson's disease is that many of the inherited forms, not all, but many of them, either directly or indirectly, have an effect on mitochondria. So either getting rid of problematic mitochondria or making sure that mitochondria function properly. You can actually trigger chemically induced Parkinson's disease by giving them a drug called NPTP. And what this does is it actually causes major disruption of the electron transport chain, and it can actually trigger high levels of reactive oxygen species production, and that can actually selectively trigger the death of dopaminergic neurons. They noticed from the 70s that somebody was making some illegal drugs in San Francisco, and a bunch of people took it and didn't realize that in the drug synthesis process there was high levels of NPTP. And I don't know, dozens of people got Parkinson's disease from that contaminated drug. So mitochondrial dysfunction and not getting rid of problematic mitochondria is an underlying theme of many neurodegenerative disorders. The best one is characterized as Parkinson's disease. Okay, so then this leads to where I got interested in autophagy. So about 20 years ago, I was working in this lab, and a lady that I worked with fruit flies, or Drosophila melanogaster, and she was studying this process that was kind of new and emerging called autophagy. We didn't know a lot about it. We knew a little bit. Most of it was discovered in yeast, but they were finding, hey, this also has roles in higher organisms, multicellular organisms, including mammals and humans. But to understand its role in an aging context, Drosophila melanogaster is a very convenient model to use because it has a relatively short lifespan, roughly 60 days. And so again, genes that are really important tend to be highly conserved. You find it in yeast, you find it in a multicellular organism like a worm or a fly or a mouse or a human. And so there's ways that you can use genetic manipulation to disrupt genes in Drosophila. In this case, we disrupted the ATG8, also known as LC3 gene, and we found that the wild type, we compare it to, this is looking at how long they survive. So as I said, anywhere from 40 to 60 days they survive. And when we mutate the ATG genes, actually flies have two of them, we mutate both, they have a much shorter lifespan. And then when we stain the brain, with ubiquitin, we find there's all these ubiquitin aggregates, and that could be a combination of proteins that would be ubiquitinated, not targeted for degradation, or it could be, for example, mitochondria that are being ubiquitinated, but they're not getting processed. They can't have the LC3 come and form that isolation membrane around them. So we get a combination of protein aggregates and damaged mitochondria that accumulate in the brains, particularly the brains of these animals, and this leads to neurodegeneration and a shortened lifespan. So then we did the opposite, using genetic techniques to overexpress ATG8, and these flies lived much longer. They had less polyubiquitination of proteins and mitochondria, and they were living like 80 days and still very spunky, perky flies flying around mating, whereas at the comparable age, any flies that were still alive were really decrepit. These were like super flies. So this was really interesting. And the overexpression was actually done only in the brains of these animals. So by maintaining autophagy at appropriate levels with age, you can actually counter some of the age-related effects that we see. So that's fine in flies. What about in something that's a little bit closer to us, at least a mammal, a vertebrate, in this case a mouse? So again, it turned out if you delete, so ATG5, you go back to a couple of slides ago, that's one of the proteins that are involved in initiating autophagy, starting the formation of that isolation membrane. So nucleation's formation of isolation membrane. So it starts the ball rolling. So if you delete it, if you completely delete it in mice, it's actually lethal. So the pups are born, but the period of time between birth and suckling is where mice require autophagy, because they have to make use of whatever fat reserves come from embryogenesis and fetal development until they can start to suckle. When you completely delete it, they can't initiate autophagy in the postnatal state, and they die. So in this case, what they did is they used some fancy genetic techniques to only inactivate the ATG5 gene in the brain only. And in this case, what we see is they ended up getting progressive neurodegeneration, so they lost brain cells. And that was associated with the formation of ubiquitin-containing protein aggregates. So proteins that misfold tend to be targeted with ubiquitin, and if they misfold and aggregate to a large extent, the only way to get rid of them is through autophagy. So these animals, when we stained, so the brown stain is staining of ubiquitinated protein aggregates, lots of it found in the brains of the knockout, so ATG5 knockout in the brain. These are the control animals. And we can we can assess various neurological functions. So we can do a stride test. So you put, paint on the feet of the mouse, and then you get it to walk across some paper. So the mouse at the top there is a normal stride. This is a knockout mouse, and it's kind of staggering all over. This is a startle test. You pick the mouse up by the tail, and it puts its legs out. This one just sits there. And then finally, it's sort of like a lumberjack on a floating log. So this is a rotor-rod test. So you put the mouse on a slowly moving rod, and it can keep up with it. And in this case, the knockout mouse just falls off right away. So that shows you if you're not capable of activating autophagy, this can lead to pretty pronounced problems in the brain. And then finally, what happens again? So this is sort of like an experiment we did with flies, but we inactivate, we see things get worse. What happens if we overexpress the ATG5 protein in mice? And so, first of all, they live longer. So the green lines are the transgenic mice expressing high levels of ATG5. And this is ATG5 expressed universally in all tissues. So This you can see in brain, lung, and heart. Okay, so these are Western blots. I know you maybe have seen these before, so it's a way to quantify protein levels. And so the darker those bands mean more dark bands means more protein. So W stands for wild type, T stands for transgenic. And this is just confirming we have higher levels of ATG5 in the brain, lung, and heart. This also correlates with longer lifespan. Now, what's this thing in beta-actin? It's what we call a loading control. It's a protein that is not expected to change in any great manner based on manipulation of ATG5. We're not manipulating beta-actin. It's a really abundant protein. And you can see it doesn't change throughout. And that's basically to ensure when we loaded that protein on an SES page gel, that we loaded equal levels of protein throughout. When we do an electroplot, which Dr. Pemberton will talk about in the next week or so, these loading controls are really important to ensure that if you see a change in any of these bands up here, it's due to the genetic manipulation, not to you loaded more protein here. If that was the case, these bands would be even stronger. So then that's just sort of a technical issue there. Okay, so yes, one quick
Are there
question.
any negative side effects associated with the overexpression of ATG?
So the question is, are there any negative effects associated with ATG? The answer is there can be. So autophagy is a process that has to be selectively turned on. If you turn on one component too much, it actually can cause a buildup of one of the steps which can interfere with further progression. So making really high levels of autophagy proteins can be problematic.
Directly, I don't think genetic manipulation is well regarded, but there's chemical activators.
For a second, I thought.
Definitely what's called rapamycin. So rapamycin is being used actually for transplantation, tissue transplantation studies as an immunosuppressant. Last 2 lectures is treat patients at a much lower level. It doesn't cause the suppression of the immune system, but it does actually cause all those examples of animal models that are stacked from 8 a.m. to 6 p.m.